Namaste students. For sure with you. I, JVN, Dr. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the transgenic plant. So, transgenic crops. So, transgenic crops is a genetically modified organisms, or we can say transgenic indicate that the transfer of a gene has occurred using a recombinant DNA technology. Okay? Generally, transgenic crops contain one or more genes that have been inserted artificially either from unrelated plant or from any other different species. So, a transgenic plant contains a gene or a gene of different species artificially inserted into its genome. The inserted gene sequence known as transgenes and may come from an unrelated plant or from a completely different species. For example, BT cotton is a transgenic cotton plant that incorporates a gene that the bacterium or the Bacillus thuringiensis or BT produced crystalline like parasporal bodies during the sporation and the parasporal morphology. The biological use of BT in insect pest control is centered on the exploitation of their crystalline protein. Especially, these crystalline proteins are found to be highly toxic in to agriculture important pests at very low concentration. So, what is the requirement of making a transgenic crop? The conventional plant breeding involves the exchange of genes between the plant of the same species or from closely related plant to produce a hybrid having a desirable trait. The crossing between, however, is limited to exchange between the same or closely related species. Therefore, it requires a long time to achieve the desired results, or sometimes a related species having the character of interest may not be found, or incorporation of undesirable characters such as the origin of the new pest, disease, incident, or genetically engineer enables the transfer of the genes more easily across the taxonomical boundaries. Okay. Transgenic plants can incorporate the desired trait more quickly and more reliable than the conventional method. So, how we can produce a transgenic plant? A transgenic plant can be produced through a genetically engineer in which the genes that codes for a desirable traits are transferred from one organism to the other organism. The universal presence of DNA, that is deoxyribose nucleic acid, in the cell of all living organisms is the basis of development of the transgenic plants. Okay, so these molecules stores the organism genetically informations in the form for its metabolic process. So the genetic information is specific of the sequence of four chemicals like adenine, cytosine, guanine, thymine. So along with the length of the DNA molecules joins the deoxyribose and phosphate group. The combined unit of the nucleotide and this base couple selectively forms the adenine and guanine and cytosine and thymine. So the genes are sequenced of nucleotide, and once the sequence is defined, the gene has no special relationship with the organism in which it is found. So the combination base of the genetic word called codon, and the genetic information is expressed in the form of the protein by an intermediate molecules, that is RNA. And the property of every organisms are determined by the protein, and it is made up of cells. The genetic code is universal code, and the DNA segments form from one organism. Okay. So the genetic engineering begins with the identification of the genes responsible for the trait of interest. The most important tools of genetic engineering are enzymes that perform specific functions of DNA. Restricted enzymes recognized and cut DNA at a specific region. Uh, and the other enzymes known as ligase joins at the end of the two DNA fragments. The use of these enzymes enables the manipulation of the DNA even in unrelated organisms. So identifying and locating the gene for agriculturally important trait is currently most limiting step in the development process. Relatively little is known about the specific gene required to enhance the yield potential. So to improve the stress, drought, chemical properties, or harvest products. So many once the gene has been isolated and cloned, uh, it must undergo several modifications before it effectively inserted into a plant. So here the selectable marker gene is added to the gene construction and to identify the plant cells or tissues have been successfully integrated the transgene. So two primary methods currently existing for the introducing the plant genome. Uh, a process is referred to as transformation. So the first device involved is gene gun. The DNA to be introduced into the plant cell and is coated into a tiny particles, and these particles are physically shot on the plant cell, and some of the DNA comes off and incorporate into the DNA of the recipient plant. The second method is the agrobacterium introduced or into the area of interest into the plant cell, and there it is further it is difficult to determine whether the new gene has been incorporated or not. So the next step is selection of the plants is then crossed and improved variety of the crop, because only few varieties of the given crop can be efficiently transformed. Next up is the process of multi-locations and multi-year evaluations in the greenhouse and the field environment to be tested of the transgene and overall performance. This phase includes the evaluation of environmental effect and food safety also. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, please write down in the comment box. I will resolve it in the problem solving session. Thank you.